So we recognize then that promoters are the initiation site of transcription. They are where all the action kicks off for this process of converting DNA to RNA. But you may have, of course, recognized that a promoter is not actually a blue box, and in fact it is rather a sequence of nucleotides, a conserved sequence of nucleotides. That is, if we were to look in different groups of organisms, prokaryotes across the board, we would see these consensus sequences. That is, if we compared in many different bacteria and looked for what happened in this promoter region, we would begin to see that the sequences would repeat. And we can look for which nucleotide occurred most frequently in each location in this region and determine a special consensus sequence that actually dictates the promoter. So it's actually a consensus sequence that is recognized by the initiation machinery to kick transcription off. And that particular prokaryotic consensus sequence is TT-GACA at the 35 nucleotides to the left of the start site, and something we call the TATA box at about 10 nucleotides to the left of the transcription start site. So these are really what are recognized by the initiation machinery as the cue to begin transcription. They are what kick it all off. So let's label these as the -35 consensus sequence, again occurring about 35 nucleotides before the start site, before what we call the +1 start site. And then the TATA box occurring about 10 nucleotides before the transcription start site. So these two regions are recognized by the eventual enzymes that are going to convert the DNA into RNA. And so in doing that, they really do kick off the process. Now, depending upon the prokaryotic organism, it may have a promoter that very, very much resembles this ideal consensus sequence. That is, this consensus sequence that was determined based on comparing everything and finding that most frequent nucleotide. So some things have a a promoter sequence that is very, very close to ideal. And in those cases, we say that the promoter is very, very strong. And essentially what that means is that because this promoter is so close to the ideal consensus, the machinery that causes transcription is much more likely to recognize and bind to this particular promoter. So what that means is that the gene that is under control of that promoter is much, much more frequently transcribed. That it gets a lot of action, right? It's going to see a lot of beginnings and readings of the gene. So a very strong promoter is one that gets transcribed very, very frequently, has a very, very close to ideal consensus sequence. But a weak promoter is one that gets read very, very infrequently, has a consensus sequence that differs a great deal from the ideal consensus sequence. That is, it doesn't have something that looks very much like TT-GACA or TATA-AT. So to really kind of understand this, I think we should do just a little bit of practice and see how good of a job we can do at recognizing strong promoters. So let's shoot on and do a little bit of that, looking at a few examples of different promoters and seeing if we can actually pick out the one that is the strongest. So keep in mind what the consensus sequence is, and in fact maybe I should just write that down. Remember that our consensus sequence is 5', and at the -35 sequence we have TT-GACA, and then the -10 region ideal is TATA-AT. And then of course down to the +1 start site where transcription actually begins. So at the +1 is where the process actually begins. So let's compare ideal to what we see in these examples and see if we can actually pick out the one that is the strongest promoter. Let's start with the top one, oh, there's a change, GACA. Okay, so the C would have been a G in ideal. And then TATA, oh, look at this, two differences. Those two would be A's in the ideal consensus sequence. So there are 3 differences from ideal, making this not necessarily a weak promoter, but weaker than the ideal consensus sequence. Now looking at the next one now, down, TT GACA, so 2 differences there. At, wow, that one is really a weak promoter, isn't it? Because it really doesn't resemble ideal very closely. So let's go down to the bottom one, TT GACA, oh, 1 difference there, the C, and then at, okay, 2 differences. So we can say with some confidence that the bottom promoter is the strongest promoter, and the gene or genes that are controlled by that promoter are transcribed far more frequently than the others that we have looked at prior to that example. So now we should be pretty good at recognizing strong promoters and knowing that one of the things that controls the rate of gene expression is the strength of the promoter. The closest to ideal, the more frequently those downstream genes are transcribed. Let's do another practice question to make sure that everybody's totally on the up and up with this process. So all of the following accurately describe either prokaryotic replication or transcription. So this allows us to bring some concepts together, the replication process and the transcription process. So let's start out with A. During replication, Okazaki fragments are formed during the synthesis of the lagging strand, but not during the synthesis of the leading strand. That's correct, right? Because Okazaki fragments are what are formed in the discontinuous or spurt-filled synthesis of the lagging strand. So this sounds like a good and true statement. We're going to go ahead and just put, okay, this one looks to be, you know, pretty accurate. So I'll put a T next to it, I guess. The next deoxynucleoside triphosphate that DNA polymerase would add to the currently synthesizing strand would be DCTP. Okay, well we know that, remember, DNA polymerase wicked fast, but also incredibly needy. And so we know that it has to have an open 3' hydroxyl to build from. So it has to either build here or it has to build here. Now, in order to also build new DNA polymerase has to have something to pair with. That is, there has to be a base to pair, a G to pair with a C, or a T to pair with an A. And what we notice here is that this cytosine doesn't even have anything to pair with. So in fact, the next nucleotide added to this 3' hydroxyl would have absolutely nothing to pair with. So this is out. This can't be the site of elongation. So instead, here, notice that guanine would very happily pair with cytosine. So the next nucleotide that could get added there would be, in fact, DCTP. So that makes a lot of sense. That looks like a good, and let's put a T next to that answer. Let's look at C. A promoter with a -35 sequence of TG, oh, let's pick out the mistakes right away. TT GACA, so TT GACA, oh man, that's pretty weak. And a -10 of at, 2 differences there too, is stronger than a promoter with a -5 of TT. Wow, 1 difference there, and a -10 of oh. This is a false statement. Check that out. So we can go ahead and say that this one tentatively is our answer. Maybe circle that because we want to look for the exception. Now, the coding strand of DNA is the same sequence as the RNA transcript, only U's replace T's. Oh, there it is again for the 555th time that we're going to remember that, in fact, the RNA does look exactly like the coding strand of DNA, only U's will be found in every place that would have seen a T in the coding strand. Fantastic. That was a fun practice question, so we can get to moving on and talk a little bit more about transcription. There's just a bit more lingo that we need to learn before we can get into first initiation, then elongation, and finally termination of transcription. So let's begin with some of this additional terminology. I've been trying not to use the word, but the region to the left of a gene or to the left of a promoter is always termed the upstream region, whereas the region to the right is always termed the downstream region. And it's easy to remember that, in fact, a promoter is always upstream of a gene, because we know that the promoter is always providing the start site for the transcription of that gene. So it makes a lot of sense that it would always be upstream. So if you're ever looking at a gene and you're trying to figure out, okay, which direction is upstream and which direction is downstream, looking for the promoter is helpful. But of course, as long as things are written with the 5' to 3' strand on the top, that's another way to recognize that a movement towards the 5' end of the coding strand is movement in the upstream direction. Movement in the 3' direction on the coding strand is movement in the downstream direction. But of course, recall that things are a little bit relative. And when we think about the prokaryotic world and the fact that many of their genetic elements are plasmids or the single circular chromosome, that we tend to see things in circles. So then you have to start to think about how the promoter dictates the upstream region of a gene. And in fact, I'm going to show you an example where we can see on a plasmid 2 different genes. And this is kind of fun, because what we notice is that the promoter for gene 1, okay, so promoter for gene 1, right there, we know that's going to be upstream of the gene. So that means that all the machinery needed to transcribe that gene, that's all going to bind here at the promoter, and then it's going to move in this direction, reading the bottom strand and making a product that looks like the top strand. So the resulting mRNA, in this case, the mRNA is going to look like that red strand because the blue strand was used as the template. But now check this. Look over here at this promoter for this gene, and we should call this gene 2. I don't know why both of those got labeled gene 1. And recognize that this is going to head, the machinery to transcribe gene 2 is going to head in this direction. And it's going to use the red strand as template, the bottom strand as template, making an RNA then that's going to look like the blue strand, which in this case is the coding strand. So the purpose of this example is to help you understand that our perspective on any kind of gene's construct is relative. And depending upon where the promoter is, that's really what dictates what is the coding and what is the template strand. And because these 2 genes are read in the opposite directions on a plasmid, we see that the coding strand for one is the template strand for another, and the template strand for one is the coding strand for the other. And so that, I hope, makes it clear that everything is really relative when reading these genes. Let me know if you have questions on that, because I would often pause here in lecture and give a moment for you guys to ask a few questions. But since we don't have that moment and I don't know your questions, please feel free to email them to me and I will be sure to get back to you on those. We're now ready to talk about the initiation phase of this process, and remembering that we can split transcription, translation, and replication down into initiation, elongation, and termination phases. So in initiation in transcription, what happens is that an enzyme called RNA polymerase recognizes the promoter. So RNA polymerase, you guessed it, is the enzyme that builds RNA using DNA template. And RNA polymerase is pretty awesome. It's a multi-subunit enzyme, which is a fancy way of saying that this enzyme has a lot of polypeptide chains in it. In fact, it has 2 alpha, sorry, that's a horrible alpha. 2 alpha subunits, a beta subunit, a beta prime subunit, finally, then a sigma subunit, my favorite, and an omega subunit. So noticing that this has 5 different polypeptide chains that make up this enzyme, we can say that it has a high degree of what kind of structure? Hopefully you're getting this and thinking, lots of quaternary structure, right? A lot of quaternary structure in RNA polymerase. So RNA polymerase recognizes the promoter, but maybe I should specifically say that our sigma subunit of RNA polymerase, which I'm showing in green right here, recognizes the promoter. Sigma subunit is fantastic because it is actually what we term a promiscuous subunit, meaning that it will associate with the rest of the enzyme only transiently, long enough to recognize the promoter and say, whoa, hey promoter, good looking, I'm going to stop here and bind to this promoter. And that is all that sigma subunit does. And once the RNA polymerase enzyme has left the promoter, sigma subunit's like, hey, I'm not so interested in this DNA anymore, and it gets the heck out of Dodge and goes off and associates with another RNA polymerase enzyme. So the role of sigma subunit in the RNA polymerase enzyme is specifically to bind tightly to the promoter. So picture RNA polymerase scanning along the DNA, just running along it like a race car, and it comes to the promoter region, and sigma subunit says, whoa, hello, and it binds tightly right there at the promoter region. And then it starts to begin to synthesize RNA using the DNA template. So the first couple of nucleotides are incorporated into the RNA at this point. So that is the first thing that RNA polymerase does, is to unwind a local section of the DNA and begin to add nucleotides that pair with the template strand of DNA. So we would recognize just a couple of nucleotides getting added right here and pairing with the nucleotides on that template strand of DNA to synthesize just a short little stretch of RNA that I'm showing by that little tiny red section of RNA. We call that the transcription bubble. Not surprisingly, we remember that terminology from replication as well, the replication bubble. Here we have the transcription bubble. Now this is where an analogy is helpful. Recall that DNA polymerase simply cannot do what RNA polymerase just did there. Notice that in fact it started cold. There was absolutely no open 3' hydroxyl to kick off the synthesis of RNA. In fact, the RNA polymerase enzyme simply just added the first few nucleotides all by itself. It didn't require an open 3' hydroxyl or a primer. So the thing that's awesome about RNA polymerase is that it does not need a primer. And that's in fact why primase was what began things on the lagging strand of synthesis in replication, because it was an RNA polymerase that could start cold. By analogy, maybe we can talk about, I don't know if this works for you, but Kristen, I know, does some cross-country skiing. She's more like RNA polymerase enzyme because she doesn't need a chairlift to get her started. But I think that Sydney had told me that she was a snowboarder, and she needs a chairlift, right, to get her started. But once Sydney gets started, she is wicked fast going downhill, whereas Kristen is steady Betty going up hills and down hills. So she doesn't need a chairlift to get her started, but once she's going, she's not near as fast as Sydney is going downhill. And that's the analogy between DNA polymerase and RNA polymerase. DNA polymerase wicked fast, but it has a lot of needs. And whereas when we recognize with RNA polymerase, it's not as fast. In fact, it's way slower, maybe on the order of 45 nucleotides per second as compared to the 1,000 that we sometimes see in DNA polymerase. But it does start cold. It doesn't require any kind of primer in order to get it kicked off. So a short stretch of complementary RNA is made, and it's usually somewhere around 9 nucleotides that get synthesized, just as I said, without a primer. So this begins that transcription bubble, begins the nascent strand of RNA that is now looking like, very much like the coding strand of DNA. Its little 5' end has just started to get made. So in fact, I might just quickly draw an extra cartoon of this to make sure that everybody is totally with me. So we've got this 5', and I'll just start the transcription bubble right there, and the 3' to 5' strand. And recognizing the RNA polymerase enzyme in there. Don't have an orange pen here, but we'll just do that. There's your RNA polymerase enzyme. And then as the RNA begins to form, it pairs transiently So inside of the RNA polymerase enzyme, it begins to make RNA. And that RNA, just about 9 nucleotides getting paired with the template strand of DNA. So we can recognize that this little end that just starts to escape here is the 5' end of the RNA, looking a lot like the coding strand, but user replacing T's. And if we were to zoom in, as we're going to do here in just a moment, you might be able to see, in fact, that maybe here there was a G on the template strand and that paired with a C on the RNA. So the blue template strand is dictating the sequence of the RNA that way. Marvelous. Let's go on and mention that as RNA polymerase leaves the promoter, we call that clearing the promoter, then the sigma subunit becomes very disinterested in that DNA, has much lower affinity for non-promoter sequences, and it takes off and leaves behind the core enzyme. And of course now you can see that short stretch of RNA pairing with the template strand. This now brings us into the elongation phase of transcription. And in that process, we recognize that 1 nucleotide at a time is added to the open 3' hydroxyl. We'll label that. Inside of here is the open 3' hydroxyl, to which new nucleotides are continuously getting added. Now we could label that on this bigger one, and you can see here how new nucleotide, a G paired with the C on the template strand of DNA. So see here the 5' end has emerged a little bit more, and we recognize again this looking like the coding strand. So NTP building blocks are added on one at a time to that open 3' hydroxyl. And this is what elongation is all about, is adding 1 ribonucleotide or NTP as the building block at a time. And this just keeps on happening, right? We just keep on seeing addition of 1 nucleotide after another, and this is why this step is called elongation. Do note that as RNA polymerase proceeds on down the gene and it leaves the promoter behind, the promoter is now accessible for the binding of another RNA polymerase enzyme complete with sigma subunit to begin another round of transcription. So as RNA polymerase moves, the previous stretch of DNA reanneals and the current stretch is melted. So the transcription bubble moves along as the RNA polymerase enzyme also moves along. In what I hope is like the highlight of your entire day, I do have cheesy animation number 553, and we're going to take a look at the process by which the RNA polymerase enzyme reads the template strand and makes that RNA that is complementary. Oh yeah, there it goes. Reading, making more and more RNA. This is the 5' end that is being exposed a little bit more, added each time, each movement of the RNA polymerase enzyme, and each addition of an NTP to the open 3' hydroxyl. Let's do it again. Oh yeah. Pretty cool. Let's talk just a little bit about termination as we head into the last phase of this process of transcription, when in fact eventually the RNA polymerase enzyme hits some sort of termination sequence. Termination can occur in 2 different ways. One of the most common ways is actually what we call a hairpin loop formation. So I'm going to draw a quick cartoon to perhaps show this in a couple of different ways. But what can occur is that as RNA polymerase hits a termination sequence, that sequence is often encoding for a region of complementarity on the RNA itself. So if we recognize RNA polymerase there, unwinding that local section, the RNA coming out here with its 5' end. And what will often occur is that there'll be a region in the RNA where there's a section perhaps of a bunch of C's and then a section of perhaps a bunch of G's, and that actually causes a region where the RNA wants to hydrogen bond to itself. And when it does that, it forms this hairpin loop. and there's this local region of hydrogen bonding in the RNA, and this hairpin loop destabilizes the RNA-DNA heteroduplex and causes the termination of transcription. Here's a zoom-in picture that comes from none other than Wikipedia. As long as you know your source is giving you correct information, this is a source you can use. However, it is never a source you would want to cite. You would want to go to their primary source, right? Notice here, though, they do show a picture of a hairpin loop where you have a region of complementarity that leads to that local structure, as I've drawn over here, that destabilizes and causes termination. So hairpin loop formation is one of the most common ways in which transcription is terminated. The other way is called Rhodependent termination. And in Rhodependent termination, there is an enzyme, actually an ATPase, that literally takes the RNA and wraps the RNA around itself and pulls. That maybe gives it a little too much character, but in fact it does. By wrapping itself around the RNA, it actually does destabilize the RNA-DNA heteroduplex and causes termination. Think to yourself why rhodependent termination might not be very common in prokaryotic cells. And I'll leave you with that thought. You can maybe think about why, based upon compartmentation, that might be the case. Have a fantastic day, and I look forward to another awesome vodcast for Friday.